Hi, I'm Christopher. In this short video, we're going to take a look at the alphabet soup of antibody classes and turn it into a usable framework. In mammals, antibody classes share the same basic design: antigen binding arms called FAB regions, and a stem called the FC region that communicates with the immune system. Today, we'll focus on four big players: IgM, IgG, IgA, and IgE. The goal is not to just memorize letters. The goal is to ask, where's the threat? Where's the antibody working? And what job does that antibody class need to do? The big idea is that isotype changes the antibody's job description. The FAB region still binds antigen. If a B cell changes the isotype, the antigen target can stay the same, but the FC portion changes. That FC region helps determine which immune tools get called in: complement, phagocytes, mast cells, transport across mucosal surfaces, and other class-related functions. So structure affects location and function. An antibody in blood, mucus, tissues, or sitting on a mast cell is not just in a different neighborhood, it is suited for a different job. IgM is the first responder. In a primary response, IgM is often the first antibody class made before class switching and affinity maturation refine the response. As a secreted antibody, IgM is usually a pentamer, 5 Y-shaped antibody units connected by a joining chain or J chain. This structure makes IgM big and gives it a lot of binding sites. Because of that, IgM is excellent at agglutination, which means it clusters antigens together. It is also a strong activator of the classical complement pathway. Since IgM is so large, it tends to stay mostly in the vascular compartment. One useful caveat: surface IgM on naive B cells is monomeric. But when we're talking about secreted IgM in circulation, think big pentamer. IgM is like an early alarm system. Not necessarily the most refined antibody, but big, loud, and very good at starting complement. IgG is the main systemic workhorse. In serum and tissues, IgG is the antibody most people think of when they think about a durable systemic protection. It is especially important in secondary responses after previous exposure or vaccination, when memory B cells respond more quickly and often produce higher affinity antibody. IgG can neutralize pathogens or toxins, opsonize microbes for phagocytes, activate complement, and support antibody-dependent cellular cytotoxicity, or ADCC. The exact mix of those functions depends on subclass and species. That matters in veterinary medicine. Additionally, maternal antibody transfer is not one-size-fits-all across mammals. Foals and calves receive minimal antibody across the placenta and depend heavily on colostrum. Puppies and kittens receive some before birth, but colostrum is still critical. Humans, on the other hand, receive substantial transplacental IgG. So IgG is the systemic workhorse, but species context matters. IgA is the mucosal guard. The immune system has to defend respiratory, gastrointestinal, and urogenital surfaces while constantly encountering food, commensals, environmental antigens, and normal mucosal tissue. Secretory IgA is built for that balancing act. It often functions as a dimer, with the J chain and secretory component that help it move across epithelium and persist in secretions. Rather than launching a major inflammatory event every time something touches the mucosa, IgA helps block attachment, neutralize organisms or toxins, and keeps threats at the surface. IgA keeps things at the surface without burning down the building. IgE is the high alert specialist. It is present in very low amounts in the serum, but it has a powerful role because it binds Fc receptors, especially on mast cells and basophils. When antigen cross-links Ig in those cells, the cells can degranulate and release mediators such as histamine. That mechanism is central to type 1 hypersensitivity and allergic disease. But IgE is not simply bad. It is also associated with responses against helminths and ectoparasite-related challenges, where rapid local mediator release and recruitment of other immune cells may be useful. The problem comes in when the high alert system is triggered by the wrong target or at the wrong intensity. Then a protective alarm can become collateral damage. Here's a quick comparison. For an early primary response, think IgM. For systemic protection and memory, think IgG. For mucosal surface protection, think IgA. For mast cells, allergy, and parasite-associated responses, think IgE. This is a first-pass framework. It is intentionally simple, but not simplistic. Species differences and subclass differences matter, especially in veterinary medicine. But these associations give you a practical mental map that you can build on later. The take-home message is that antibody classes are not alphabet soup. They're different tools. The FAB region helps identify the target, but the FC region helps determine what kind of immune response follows. Structure influences location, and location predicts function. IgM is large and early. IgG is systemic and versatile. IgA guards mucosal surfaces. IgE sits on a high alert pathway tied to mast cells, basophils, parasites, and allergy. The best antibody class depends on where the threat is, how the antibody is built, and what kind of immune response is needed. Thank you for watching. I'm Christopher Lee, and what you know today protects everyone tomorrow.